All right, the first thing we're going to do is talk about a guy named Gregor Mendel. And we don't usually talk about a lot of folks, but he's super important because he gave us information about how traits are inherited. Okay, so he used pea plants because those are very easy to, they flower very specifically. The turnaround time is very quickly, like they grow very fast. So they were just pretty ideal for them to, for him to use as an experiment. So prior to Mendel, so this is before Mendel, everybody thought that traits were blended. So if your dad was 6 feet tall and your mom was 5 feet tall, you'd be 5.5 feet tall, right? Or if a flower was red and another flower was white and you crossed them, then you would have pink flowers, which for some things they are. But they thought that everything was what they call blended inheritance. Okay. However, Gregor Mendel, because of his studies, came up with a different hypothesis. Okay, so he used these pea plants, and what he did was he got true breeding peas, and basically that just means these plants will always produce offspring that look like their parents. Okay, true breeding will produce offspring that look just like parents. And he did this by inbreeding the pea plants. So then he took a pea seed color. All right. So you have green. All right. And then you have yellow. So he took a true breeding green plant and a true breeding yellow plant and he crossed them. So in other words, he mated them. Now, if the blended theory was correct, you'd have some sort of pea green, like a very yellow green looking pea. But what he discovered was that when you crossed these true breeding plants, you ended up with all yellow seeds. And Mendel was a little bit confused, like you would think, again, that you'd have some sort of yellow-green hybrid. So what he thought he was going to get was something that was a little bit yellow and a little bit green. Okay? But he ended up getting this yellow pea plant. So this right here proves that the blended inheritance theory is not correct. But he did not stop there. He then allowed these particular yellows to self-propagate, in other words, to self-pollinate, so that they were genetically identical. And what they found was that some of them were yellow, but then all of a sudden, seemingly random, some of them were green. And so this led him to the conclusion that some traits are dominant. So some traits are dominant and some traits are recessive. So dominant is, he says, is passed down unchanged. And a recessive, he says, can disappear in hybridization. And what does that mean? Well, let's look at the actual genetics now behind these pea plants. If yellow is dominant and we take a true breeding dominant plant and we cross it with a true breeding recessive plant, we are going to end up with a hybrid that has one dominant and one recessive. This is the F1 generation, all right? When we take this hybrid and we cross it with itself, what we end up with is we take Y, that's okay, Y, and we cross it with Y, big Y, little y. We are going to get 2 true breeding, 1 dominant, 1 recessive, and 2 hybrids. Now, these hybrids are going to look exactly like the parent. So you won't be able to tell the difference if this hybrid is big Y, or big Y, little y. You won't know. But you know for sure that if it's green, it is 2 little y's. So let's get some vocabulary to go with this. All right, let's get a little vocabulary to understand this a little better. All right. My first example, this right here or this right here, 2 of the same size letters. This is homozygous. This means that homo means the same. And this means that they have the same size letter, essentially. All right, we can call these true breeding. All right. Now, if I have a big Y and a little y, we call this heterozygous because hetero means different. We can also call these guys hybrids. All right. Each one of these letters is an allele, which, if you don't remember, I'll just write a quick definition down here. Allele is a form of a trait. We have 2 alleles per trait. Remember, 1 from mom, 1 from mom, and 1 from dad. So when I give you the alleles, so if I show you each allele, then I'm giving you the genotype. So the genotype is the genetic makeup of an organism. Again, we're just doing some vocabulary. Genotype is the genetic makeup. The phenotype is its physical appearance or what it looks like. All right. So if we take Mendel's cross of 2 heterozygous individuals, then what we find is that we end up with, if we're just looking at phenotype, we are ending up with 3 that are yellow. And the reason is because if you have 1 dominant allele, you show that trait. All right. So again, up here, if we go up here, I'm just going to shift a minute. Dominant and recessive. If you're dominant, you only need 1 allele to show the trait. If you're recessive, you have to have 2 alleles to show the trait. So phenotypically, if I'm just looking at physical appearance, a homozygous dominant and a heterozygous individual are going to look exactly the same. talk about the fact that some traits are dominant, this is actually called the law of dominance. Basically, this means that if a dominant allele is present, it will be expressed. So I'm going to go up here. Law of dominance. If a dominant allele is present, it will be expressed. Now, we are going to find out, or as you go through your choice boards, you're going to find out this isn't always the case. But again, we're just talking about Mendel and his pea plants, okay? And that was pretty revolutionary, by the way, because remember before they thought that they were blending. And there are some instances where things are blended, but we're not going to get into that right now. All right, now let me take you up here because I actually have already done a Punnett Square. That's what this thing is called. Now, this is a monohybrid cross. That means I'm only looking at one trait. Mono means one. So I'm only looking at seed color in this Punnett Square. Now, the significance of a Punnett Square is that the Punnett Square gives you the probability that a trait will arise. Okay? In other words, if I cross two yellow peas that are heterozygous, the probability is I have a 75% chance that they will be yellow and a 25% chance that they will be green. Okay? So that is because, remember from my cross right here, this one, and this one are all going to appear green. And if each one of these squares represents a proportion. If I have 4 proportions, that's 25% each because the entire Punnett Square always has to equal 100%. Okay? So if I have 4 squares in a Punnett Square, then each one of those proportions is 1/4 or 25%. Now we'll get up into bigger ones where if you're doing 2 traits, you're going to have 16 squares. That means each square is 1/16, and you just kind of multiply them all up or add them all together. They're percentages. Okay? So let's try to do let's go back. All right, so how can we even do a Punnett Square? How do we know that this Y allele is going to go by itself and it's not going to take anything else with it? How do we know? So Mendel proposed the law of segregation. And basically, the law of segregation says that alleles separate equally. Oops. Into gametes. There is equal likelihood of an offspring to inherit either allele. All right, so what does that mean? That means if a dad is YY, if the male plant is YY, the gametes are going to be, here's his little sperms, are going to be big Y and little y. That's what he has to offer. Okay? So big Y and little y, and that the chances of an offspring inheriting either one of them is equal. All right? So this goes actually along with then the law of independent assortment. And basically what this says is that genes aren't inherited together. So every allele is inherited independent of any other. So alleles are inherited independent of each other. Now, again, we know that this isn't always true. But this is Mendel again, so this is way back. So he doesn't necessarily know that that's true. However, as you can see, we have 3 laws that Mendel gave us from his experience. The law of dominance, so if a dominant allele is present, it will be expressed. Law of segregation is alleles separate equally. And so there's an equal chance that the offspring will inherit either allele. And then the law of independent assortment states that alleles are inherited independently. So if we go back, another one of Mendel's experiment he liked to do was he liked to do a smooth or wrinkly. Okay. And what he learned was that wrinkly, so wrinkle is recessive and smooth is dominant. So if we take a pure breeding, wrinkled, smooth, and then we have our little wrinkled, and then we cross them, they are all going to be S, big S, little s, because all this one has to give is a big S, and all this one has to give is a little s. Now it gets more complicated. So then again, just like with our yellow and green, if we want to cross one of each, we're going to self-cross. Then we'll end up with, I always put the big S first, but that confuses some people. So like on this one, if you're putting this letter first here, you could always, or the little s, you can switch them. It really doesn't matter. To me, it just makes more sense to keep, if you have a dominant allele present, to keep it. So these phenotypically, we're going to have 3 smooth or 75% smooth, or 3 out of 4, or 3 are going to be smooth and 1 is going to be wrinkled, if you're doing a ratio. Now, what if we were given a smooth plant? What if we were given a smooth plant? So here you go, you have your smooth, but you have no idea if it could be big S, or big S, little s, we don't know. So the best way to do that is to do what's called a test cross. And that just means we are going to cross an unknown with a recessive phenotype. Now, we know that if you have a recessive phenotype, that all recessive organisms or all recessive traits are homozygous. So we know that our wrinkly, that if we're going to cross this unknown with a wrinkly guy, he's going to be little s, little s. That's a known. We now know that. So in order to do a test cross, we have to do 2. So if the smooth one is big S, little s, and we cross them, we should all get nothing but smooth. All right? So these are all smooth. But if we cross them, and we get some that are wrinkled, we know that that parent was heterozygous. So that's why sometimes we need a test cross for that. Okay?